10 pages. In readings? Yep. Oh, okay. Maybe mine says, is it this, the excerpt? Yeah. What? It probably is though.
All right. So today we're going to continue our discussion of ways that we balance our electrolytes, so basically our ions, using our kidneys. So when we left off before break, we were talking about aldosterone. That was our last hormone that we spoke about. So you go through this list and check off as many of these as you remember being true things about aldosterone. All right, seeing some answers coming in. Take another minute or so, and then we'll go through and keep going, thinking about our hormones. We'll do a little more talking about aldosterone a little bit today. All right, I'm gonna flip through and take a look. So put in your final answer. Okay, so let's go through it. Pulled up a couple of pictures for us from last time, but let's discuss. Yes, okay, so most people got this first one. So aldosterone, one of the things that's going to be driving all its actions is that sodium potassium pump on the basolateral membrane. So that part that's facing the blood. So this is what's going to drive the gradients of the ions it controls. So one of the ways it affects your body is by making more of them, putting them on that basolateral membrane. Okay. What we've been focusing on first is how we adjust our sodium levels. So yes, aldosterone is associated with sodium reabsorption. And the way we can do that is having a sodium channel that allows sodium into the cell on that apical membrane. And then that sodium potassium pump on the basolateral membrane is going to let it get into the peritubular fluid and then into the blood. The way we get to producing aldosterone in the first place, aldosterone is coming from your adrenal glands. So we have a whole big signaling pathway involving renin and our two types of angiotensin, our renin, angiotensin, aldosterone system, getting us to the production of this aldosterone, right? And the place that we're going to see aldosterone today is it's also going to be helping control our potassium balance. So we'll see that it regulates potassium, but because of the directionality of this sodium potassium pump, right? We're basically like trading sodium and potassium. Our potassium is going to go the opposite way. Now, it is in the distal tubule, but not the proximal tubule. Proximal tubule was unregulated reabsorption, so we don't have any hormones affecting that proximal tubule. Distal tubule, yes. The other place we saw this is the collecting duct. Okay. It's not aquaporin 2 that aldosterone is doing. That was ADH, our antidiuretic hormone. So aldosterone is about the sodium, and now we'll see about the potassium. And the effect of aldosterone on water reabsorption is only a secondary effect, because after you reabsorb all that sodium, then your ADH might be triggered, which would directly allow for water reabsorption. So ADH would be that last. Okay, so these are those pictures that we can see for the aldosterone related content. So here on that basolateral membrane, if you were struggling to picture it in your head, right? So in the presence of aldosterone, here we see that aldosterone in the blood, we'd create those sodium potassium pumps so that we can pull sodium into the peritubular fluid and ultimately into the blood. Okay. Here we see those sodium channels that it's putting on the apical membrane. And today we will also spend some time noting that we have potassium channels, but we'll see that potassium is going to end up going the opposite direction. All right, sodium coming in, reabsorbing into your body, potassium going out. And here we have that RAS system, right? So we have the production of renin by your juxtaglomerular cells in the kidney. Okay? And what renin is doing is converting the inactive angiotensinogen produced by your liver into angiotensin 1, which gets broken up a bit more by this ACE enzyme embedded in the wall of your blood vessel to angiotensin 2, which is what sends the signal to your adrenal gland sitting on top of the kidney to produce that aldosterone. So that is what we had finished up or looked at before break. Okay. So we were thinking about that renin system, that RAS system, as we were finishing up on Wednesday. So we want to touch back just for a second on that renin stuff to make some notes about when and why we would release renin. Okay, so for our renin, we know that this is coming from this juxtaglomerular apparatus. So what we're talking about, right, is this point where we have our nephron loop, right, coming back up, right? So the end parts. So here we have like the end parts of the loop of Henle coming back up, and it's touching that afferent and efferent arteriole going into and coming out of the glomerulus. And we have some associated cell types, right? So here we have macula densa cells right here on the nephron loop. Here we have granular cells on that arteriole going into the glomerulus. And we had mesangial cells. We didn't discuss them, but we didn't discuss them as part of renin, but we discussed them when we were talking about filtration barriers for the glomerulus. Our mesangial cells are like over here. So that's our juxtaglomerular apparatus that we were referring to. Okay, so renin is coming from there. And if we want to release renin, what we are sensing basically is changes related to like blood flow, blood pressure, plasma volume. Okay, so one of the reasons we might want to release aldosterone, right? When we release aldosterone, we reabsorb a bunch of sodium, right? That's the direct effect of the aldosterone. That sodium in the blood, your basically chemoreceptors, those osmoreceptors in your brain, sense that additional sodium input, and their response is to release ADH, that antidiuretic hormone. And this is all in that flow chart from your book. When we release ADH, you reabsorb a bunch of water by inserting the aquaporin 2 on the apical membrane in those same regions, distal parts of the nephron, so the distal tubule collecting duct. Reabsorb water, bring plasma volume up, bring blood volume up. Okay, so we want to release renin when we ultimately want to bring your blood volume up. Okay, so for renin release, we're thinking about things that are indicating to us that we don't have all that much blood volume, right? So we might notice this if we have basically less blood coming in, right? So our afferent arteriole brings blood to the glomerulus, right? So if you have less pressure in that afferent arteriole, that's because you've got less blood coming in, right? Which might be an indicator of less blood volume in the first place. This would also tend to decrease that glomerular filtration pressure because that pressure is really leading to the glomerular capillary hydrostatic pressure. Just linking it all together with some of our other concepts here. Another thing we might notice is renal sympathetic nerve activity. So we're thinking about how this relates to like our baroreceptor reflex, right? Which was when we talked about resistance in the vasculature, right? Our baroreceptor reflex was what's helping us adjust that tension and really that resistance in our blood vessels. If you like suddenly stand up, you have a sudden drop in MAP, right? You want that baroreceptor reflex to kick in, clamp everything down, bring your blood pressure back up so you don't faint when you stand up, right? This also affects the kidneys. So that sympathetic nerve activity, in this case, going directly to the kidneys, is a stimulus itself for renin release. And the final part is that because this end part of the nephron coming back up, right, is right here in the juxtaglomerular apparatus, touching everything else. This means we can also sample what's in the distal tubule filtrate, basically, right? Because that's where that fluid is going right there next. If we sense a decrease in the sodium and the chloride in the distal tubule filtrate, that's a kind of like sideways measure of having a lower flow kind of through that distal tubule, because no matter how much fluid is in your nephron, that ascending loop of Henle is pumping sodium into the medulla, right, to keep that osmotic gradient so that you can keep reabsorbing water. So if you don't have enough fluid going into your nephron, if you didn't have enough sodium going in, you'd still be pumping sodium out because you got to, it's active transport. So you'd end up seeing really low levels passing through these final parts of the nephron. Right. So when we do that, we are able to release renin. And all of this is kind of going to end up being an adjustment on blood pressure. So here we can see that if your mean arterial pressure drops, right, this is triggering the baroreceptor reflex. So that's that sympathetic activity kicking in, which is going to tell those juxtaglomerular cells to release renin, right? We have that drop in afferent arterial pressure, which really would be due to drop in mean arterial pressure or drop in plasma volume. Same thing, tells you to release renin. And here's that slightly more complicated pathway related to the concentration of sodium and chloride ions in those distal tubules and those distal parts of the nephron. So if you have a low glomerular filtration rate, so not enough fluid coming into the nephron, you're still reabsorbing sodium and chloride regardless, because we have that active transport in the loop of Henle. So we end up with low amounts as we pass back by that glomerular apparatus, and we can release renin. This is all with the goal of bringing that blood pressure back up, bringing that plasma volume back up. So that's one direction for our sodium signaling, right? So when we signal, okay, we have a low blood pressure, One of the ways we're going to fix this is we're going to reabsorb extra sodium so that we trigger our brain to reabsorb water, right? We think about water basically following solutes, right? That happens a lot, right? You have solute reabsorption, and then you have water reabsorption following it. Okay? In the reverse direction, we're going to have our third and final hormone or hormone pathway, atrial natriuretic peptide. Okay, so this is again going to be related to our sodium because that's what we're focusing on first. Then we'll touch base real quick on potassium. Okay, but we've got a different reason for this now, basically. So ANP, it's coming from your heart, right? So that atrial part, right? This is why our heart was a secondary endocrine organ way back when we did the endocrine system, right? So we got our heart and I got our atria on top, right? So that atrial part is referring to the atria of the heart. Okay. So we're going to release ANP when we sense that those atria walls are distending, right? And so by distending, we mean changing shape. So we kind of mean stretching out when we say that these walls are distending. This is a signal, right, that you've got lots of blood returning to the heart, right? If you've got extra fluid in there, that's kind of what would swell the atrium, right? If you have lots of blood returning to that right atrium, that is what might stretch that wall out and impact the receptors there. Okay. Now that is going to be related to other things going on in your body, right? Because if you have more blood returning to your heart, right, and you just have like extra volume of blood and fluid in your body, right? We also know that, right, if you have more end diastolic volume, right, which is another measure of the blood coming into the atria and filling there, right, you pump more blood out to the body and your cardiac output at the end of this, right? Not retesting you on that, but we're just kind of linking these concepts back together, right? So this is all a signal that you've got like extra blood coming in, which means you've got extra blood going out of the heart, which means that by the time that blood gets to the kidneys, right, this extra blood is going to tend to increase your glomerular filtration rate because it affects that capillary hydrostatic pressure in the glomerular capillary, right? So it's doing the opposite thing of our drop in blood pressure. Right now we've got like extra. We've got extra pressure, which means we've got extra filtration. Okay. So your afferent arteriole in this situation, right, instead of being kind of like small and loose and not full, right now we've got lots of pressure building up in that afferent arteriole, right? Which usually, we know that when we have stretch there, one of the things that we saw happening are myogenic regulation, as that would clamp back down kind of naturally. But this still does have an effect here. Okay. This is also going to be related to constriction of the efferent arteriole. Okay. So what we're going to do when we have this ANP around is we're going to try to decrease our sodium reabsorption, right? So increasing sodium reabsorption with our aldosterone ultimately resulted in releasing ADH and increasing water reabsorption. So what we want to do now, right, is the opposite. We want to close those sodium channels because we'd like to stop reabsorbing so much water, right? We really would prefer to increase our sodium excretion, right? Increasing sodium excretion and therefore not having that sodium in the plasma means we won't absorb quite as much water, which means we can kind of protect our heart ultimately, protect stress on the heart. Okay, so we are about to go to a flow chart, but just checking. So we just said, so if our plasma volume is increasing, are we going to see ANP, aldosterone, or ADH being produced in order to maintain our balance here? right, take another couple seconds to pick one. All right. Great. Yeah, we were talking about our ANP. So that's it. So going through this flow chart, right? What we're doing is noticing that we have a rise in plasma volume. So plasma is the watery part of your blood, right? It's that extracellular fluid surrounding your blood cells, right? So that extra volume, mostly water, it's stretching out the walls of the atria, which is our signal to release ANP. Okay, so this has a tendency to increase our glomerular capillary pressure, increase our glomerular filtration rate. And as we have that extra filtrate move through, one of the things we might do is get rid of extra sodium if we just have like this fire hose going through. So that's just kind of a downflow effect of all of this in the kidney. Okay. But as we continue to adjust this, right, sodium excretion, right, we would eventually readjust that plasma volume back down. Okay. This interplays with the aldosterone system here in a way that if we have our plasma volume going up and we're releasing ANP, at the same time, we're going to be decreasing renin secretion. So like the opposite thing is happening with our renin and aldosterone. So we're decreasing aldosterone production, which means we're not reabsorbing as much sodium anymore, right? Sodium was coming in through sodium channels that aldosterone put there. We're turning off this aldosterone signal at the same time that we're turning on this ANP signal. So we are no longer reabsorbing as much sodium, right? That sodium reabsorption went down. So our overall effect of these two things together is that more sodium gets excreted. As we excrete all this sodium, right, we're going to probably reabsorb less water at the same time. So finally, I'm going to briefly mention our potassium. So we talked about water. Water is super important for the kidneys. Right? So we talked about water reabsorption unregulated in the proximal tubule. We talked about water and its relationship to the renal medulla and that medullary osmotic gradient set up by the loop of Henle. And we talked about its relationship to antidiuretic hormone in the distal tubules and the collecting duct as we determine how much water we want to reabsorb or how much water we want to leave in the urine. And then we tacked on talking about sodium and how that's related. It's also getting unregulated, reabsorbed in the proximal tubule. When we get to the distal tubule and collecting duct, if we want to reabsorb more sodium, take it in, we're going to be producing aldosterone to do that. We're going to have a domino effect where we then also absorb more water using that ADH. And then we've just added on ANP. You want to get rid of your sodium, you're going to be releasing ANP. Okay. So our potassium balance is going to be a little bit simpler. We're really only going to be thinking about that aldosterone for our potassium balance. So we do have some new words. Right, so we had hypernatremia for high sodium. Right, now we have potassium as an ion, it's that K plus, right? So we have the emia ending for blood. We have hyper for high. And here we see that potassium in the center. So hyperkalemia refers to high plasma potassium, and the opposite, low potassium in your blood, is hypokalemia. Okay. So just like sodium, potassium is important for your cells, particularly your excitable cells. So we're thinking nerves, we're thinking muscles, including the heart, right? We remember all these ion gradients are really critical to things like action potentials. So we got to have our potassium right, just the same as we have to have our sodium concentrations correct. Okay. So here's a rundown of what is happening to our potassium in each of the parts of our nephron. So right at the front in our glomerular capillary, potassium's coming into the nephron, into Bowman's space freely, meaning there's no extra barrier for potassium. However much potassium you have in your plasma, and that's how much potassium is coming into the nephron in the filtrate. Okay. In the proximal tubules, we have potassium reabsorption. So it's coming out of the filtrate, out of the proximal tubule, and it's going into those peritubular capillaries. So going back to your bloodstream. So that's our reabsorption. We don't do any regulating here. That proximal tubule is our mass reabsorber. Okay. Now, when we think about the distal tubules and the collecting ducts, here is where things are going to get a little trickier, right? So here's where we can regulate things, and we can actually have two different actions going on in the distal tubules and the collecting ducts. So some reabsorption is going to happen and some secretion is going to happen. So you can pull potassium back into your blood sometimes, and you can secrete it. So you can take it from those cells lining the distal tubule and collecting duct and secrete it, put extra potassium into that filtrate as it's on its way out of the body as urine. Okay. So it's specifically that secretion part that is regulated by aldosterone. Right? So aldosterone we talked about as regulating sodium reabsorption. Aldosterone at the same time regulates our potassium secretion. Opposite directions, because this is all driven by that sodium potassium pump, where we have opposite directions for sodium and potassium movement. Okay, so aldosterone is regulating specifically the principal cells in the distal tubules and the collecting ducts. It's just a type of cell. Okay, so when you have potassium in the plasma, if you have extra potassium in your blood, that tells your body to directly release some aldosterone, right? So it's a trigger for aldosterone release. If you keep having more and more potassium, you're going to release more and more aldosterone as your body tries to adjust. Okay. So here we see a picture of what's happening with potassium in the proximal tubule. So that was up at the beginning, right? We actually don't know how it's coming in on the apical membrane in this picture, but we know that it is because it's moving out through some potassium channels across the basolateral membrane. And ultimately our blood flow would be over here, right? So it would go through that way. It also squeezes through our leaky tight junctions sometimes, okay? So going into the peritubular fluid and then into the peritubular capillaries that way as well. So that was that unregulated reabsorption in the proximal tubule. Okay. Now we're looking at the difference, what's going on with the secretion, right, in the principal cells when we look at the late distal tubule and the collecting duct. So secretion is the opposite direction, right? So that secretion, we see potassium coming in from this side of the cell where we would have our capillaries and our blood flow. So it's coming into the cell through that sodium potassium pump, and then it can go out channel on the apical membrane into the tubular fluid, which continues through the collecting duct and eventually out the kidney as urine. Okay. So this is just the same process, or all of a piece with that aldosterone regulation of sodium, right? So when we have that aldosterone, right, it affects the sodium potassium pump. We saw that when we talked about how it's creating sodium reabsorption, right? But now we're focusing on the fact that potassium is moving in the opposite direction from the sodium. So the potassium is moving into the cell, right? Into the cell while the sodium was moving out of the cell, right? And then when we open that channel on the apical membrane, right, the potassium moves out and into the tubular fluid. So this is all because we have aldosterone present in that peritubular capillary in the blood. That's our signal to our principal cells to do this, basically. So aldosterone is doing multiple things, right? So it was increasing sodium reabsorption when we think about sodium balance. We know it's coming from the adrenal cortex, released by that RAS system, starting with that renin release, right? At the principal cells of the distal tubules and collecting duct, we are simultaneously reabsorbing sodium and secreting potassium, all because we are increasing these sodium and potassium channels on the apical membrane and the sodium potassium pump on the basolateral membrane. So those are our hormones in full that we're interested in, right? So basically what we're thinking about, all of it going on in the late distal tubule and collecting duct. We want to know what ADH does. So antidiuretic hormone involved with water reabsorption. We want to know that aldosterone is responsible for both sodium reabsorption and potassium secretion. And we want to know that ANP is acting on sodium. It's doing the opposite thing than aldosterone. So it is causing increases in sodium excretion. So we're not reabsorbing it, we're excreting. So just those three. Which brings us to our final topic, which is going to link together our lungs and our kidneys. So our final really important job of the kidney for this unit is the fact that your kidneys help balance your blood pH, okay? So we're gonna be pulling both from the respiratory chapters in your textbook a little bit at the end that we didn't do at that point, and linking that up now with the pH stuff from the kidney unit. Okay, so we're going to think about what pH is, like how we have blood pH. We're going to think about how we can disturb that pH, right? So how do we get it in a bad range? And then how can we fix a pH that's outside of our desired range, either using our lungs or using our kidneys? Okay. So a reminder about pH. So we have a pH scale going from 0 to 14. Okay. So this is ultimately in our body's going to be regulated by respiratory and renal systems. It's really important from a physiological perspective because chemicals behave really differently in an acidic environment versus in a basic environment. So we've got everything nice and adapted for operating within a normal range. And our normal range here for our pH is 7.35 to 7.45. So we got a little bit of a wiggle room. Anything lower than this, right? We're not picking 7 directly in the middle like you might in a chemistry class. We're going to be thinking about relating this to the normal range for a human body. So anything below 7.35 is too acidic, right? So that's acidosis, right? So anything from 0 to 7.35. Anything too high, alkalosis, right? So alkalosis, too high a pH. And these are related to hydrogen ions, or protons, right? That's what the H is here in our pH scale, right? So if we have our normal range, we've got a normal amount of hydrogen ions. We have too acidic an environment. That means we have too many hydrogen ions. We have an alkaline environment, so like a basic environment, we don't have enough of those hydrogen ions when we're measuring. Okay. Now, pH changes in your blood are really bad. So if you get above 8 or below 6.8, this can be fatal like that, right? This is not good. This is because our proteins are all these big foldy molecules that are shaped and sticking together because of the properties of those amino acids, right? We have charged ones, we've got all sorts of different chemical and physical properties to those polypeptide chains to amino acids. And as soon as you change the pH that they're in, got a bunch of extra hydrogen ions floating around or not enough hydrogen ions floating around, that protein's not going to work right anymore. And our proteins, right, are making up our transporters in the cell membrane. They're making up enzymes doing stuff, right? They'd be doing all those clotting factors. They're doing practically every job in our cell, right? So if you mess with the proteins, you're basically messing with everything, right? So if you change the shape of the protein, you change the function of the protein. Change in shape, referred to as a conformational change. Conformation is just the shape of the protein. In terms of what you would expect to see in like a patient in these different conditions is if you have acidosis, right? So your pH is a little too acidic, but you're not to the fatal within seconds point, right? We got a little bit of wiggle room there, right? Between 7.35 and 6.8, we've got a little space. What you're going to end up seeing is a depressed central nervous system, right? So we're messing with like their action potentials, right? They're not going to be firing off as frequently as they should, right? It's going to be one of the immediate effects. In alkalosis, it's going to be like hyperactive central nervous system. So you're going to be firing off your action potentials when you don't want to. If you mess with your nerves, really not good for your body. Okay. So how do we keep this in check? Our kind of first, one of our lines of defense, I'll say, is going to be buffers in your blood. Right. So a buffer, basically like a sponge that's going to just kind of be there and able to soak up, release your excess acids or bases as necessary, right? So one of the buffers that exists in your blood for the pH is actually hemoglobin itself. So hemoglobin, talked about, is binding oxygen, right? So binding oxygen, releasing oxygen at tissue. Ideally, we talked about the relationship between hemoglobin and oxygen and what can affect it. And when we talked about the Bohr effect, one of the effects of how we could shift that hemoglobin oxygen dissociation curves was pH itself. So here's what's happening with the hemoglobin and those hydrogen ions. Right, so we've got reversible reactions, right? So here we have our oxyhemoglobin, right? So hemoglobin bound to oxygen. And here we have our deoxyhemoglobin. So hemoglobin and oxygen separate, right? And it happens to be the case that deoxyhemoglobin has an affinity for hydrogen ions, right? So when you have deoxyhemoglobin, you can tack on a hydrogen ion and bind it on to your hemoglobin pretty readily. So we can kind of adjust how much oxygen is bound to hemoglobin if we want to get ourselves some hemoglobin to soak up hydrogen ions. So that's why part of why we had that interplay between pH and the affinity of hemoglobin and oxygen. But that's not the main thing that we're going to be thinking about when we think about regulation of blood pH. The biggest buffer and our most important buffer and what the kidneys are going to really be changing is bicarbonate. So bicarbonate is also a buffer acting as a chemical sponge. The way that's working, right? Here we have our bicarbonate ion. Right? Here we have our hydrogen ion. Okay. Together, we have a reversible reaction forming our carbonic acid. And carbonic acid can also split up in a different way into CO2 and water. Okay. So this is really going to be forming that link between our lungs and our kidneys, right? When we think about our lungs, right, we're really thinking about how much carbon dioxide are we breathing out? When we think about our kidneys, we're going to be thinking about how are we adjusting our levels of bicarbonate. So we can basically attack the system kind of from two ends, depending on what's going wrong, all to help us figure out how to get our appropriate number of hydrogen ions for our formal definition of pH. So to be in our normal range, and our normal range of 7.35 to 7.45, we're going to find that rather than looking directly at hydrogen ions, it can be more helpful to look at ratios of more physiologically adjustable substances. So we're going to be paying attention to the ratios of bicarbonate to carbonic acid and to carbon dioxide, right? So what we want a 1 to 20 ratio, and we'll take a look at that in a second. That's going to give us our normal pH. And these things are all related together by these chains of reversible reactions. And any way we shift that is going to shift our pH. Okay. So technically, this is going to be governed by our Henderson-Hasselbalch equation. We will not be doing the math itself, but the way you get the pH here is you take 6.1, and you add up the log of that bicarbonate. And if we want to relate it to CO2, we can put that CO2 on the bottom. Okay. These are the two things that we're going to be tracking as we go through our renal effects on pH, as well as our respiratory effects on pH. So we're thinking about bicarbonate ions, and we're thinking about carbon dioxide. So we've made ourselves an equation that gives you pH relating to those two things. Okay. So we want this 20 to 1 ratio. 20 bicarbonate ions weigh more than carbonic acid, and as a result, more than carbon dioxide, right? So that's what we like. So we can have problems if the respiratory system messes up our carbon dioxide concentration, right? That's going to shift our pH, right? Whether we have more, whether we have less. And our kidneys, through the action of, we'll see some of these regulated parts of our nephron, we're going to be changing bicarbonate ion concentrations. So we can also mess stuff up from the other end or fix stuff up from the other end. We'll be taking a look at both those pieces. Okay. So we're going to start with respiratory stuff. What we're really just thinking about first is problems, kind of. Okay. So if we have a respiratory problem with our acid-base balance, right? So a respiratory acid-base disturbance, that means that something about how our lungs are functioning, really usually how fast we're breathing, is messing with our carbon dioxide side of this equation. Okay. So you can end up in respiratory acidosis if you have an increase in carbon dioxide. So carbon dioxide, ultimately, you want to think of as acting as a biological acid, right? So extra carbon dioxide is going to make stuff kind of acidic. And we've mentioned that as an aside before, but now we're really thinking about it as we're thinking about acidosis. Okay. So if you breathe too slowly, right? You don't breathe off your carbon dioxide, you may end up with a buildup of carbon dioxide in your blood, causing respiratory acidosis. If you hyperventilate, you're going to be breathing out lots and lots of carbon dioxide, possibly extra carbon dioxide, in which case you might find yourself in respiratory alkalosis. So if we think about this, thinking about that balance between carbon dioxide and bicarbonate, say you're in the situation of respiratory acidosis. So you've got extra carbon dioxide in your blood. What might you do if you wanted to fix that? right? So if you want to put the pH back to normal, and you're going to do it with the kidneys, what would you do with your bicarbonate ions? Where would you want to move them? Okay, take another couple of seconds. Take a look. All right. So let's think about this. So this is how we are going to adjust. So we're going to have like renal compensation for respiratory acidosis. So what we're talking about here in this situation is our problem is that we have too much CO2 here in our blood, right? So that's what respiratory acidosis is. We've got extra CO2. What we're going to want to do is we're going to want to add our buffer on, right? We're going to want to soak up the extra pH with extra bicarbonate ions. Okay. And the way we can do that, right, to get extra bicarbonate ions to soak up that acidity, so we're going to need to pull them into the blood, right? So that direction, pulling them into the blood, is reabsorption. So we're going to want to increase our reabsorption of bicarbonate in this situation. Both secretion and excretion would move us the opposite way, right? Secretion would put us into our renal tubule, into the filtrate, and then excretion would get rid of those bicarbonate ions completely from the body. So they'd have the opposite effect. So they'd kind of make this situation worse. Okay. So we will pick up here next time.